Yeah, in that line, can you all hear me? In that line, the experimental design is very important to have a proper, appropriate experiment. Can you all hear me?
Yes, sir.
Yes, sir.
Okay, right. Okay, fine.
Yes, sir.
Yes, fine. Then on that line, we started with a topic called as an introduction. Under the introduction, we said there are some principles of experimental design. Some of the principles that need to be followed to do the experimental design. So under that, what are the principles that we have already covered? Randomization, replication.
Replication.
Yeah, randomization and replication are two principles. The third principle that is also equally important principle as the other two principles is called as control of experimental error. Control of experimental error. Actually, in the last semester, I have explained what is error. Can somebody in very short form explain what is error? Can somebody explain what is error? If you are not going to tell me exactly, you see I have the list of names which you can see, right? So randomly I will pick up and ask a question from that particular person, right? If I don't hear anything, then I will assume that that particular person is absent. Do you all agree? Whether the answer is correct or wrong, you should be able to answer. Then only I will understand whether you all have got it correctly. Do you agree with that policy? Do you agree with that policy?
Yes, sir.
Yeah. So when I ask questions, please answer. Unmute your microphones and then you can answer. That is the best way to have a better interaction in teaching, right? So the third principle is control of experimental error. What do you mean by experimental error, first of all? We are going to control the experimental error. When you say experimental error, usually we do some errors. Personal error, for example. When you do experiments, we do personal error. Some errors may be due to instruments. Instrumental error, isn't it? When you have a improper tape measuring plant height, suppose let's say, if the tape is not in good shape, good scale, then it is an instrumental error. So likewise, there can be any type of error. So in an experiment, if you control all these personal error, instrumental or the measurement error, whatever the error, you control all type of error, but still there is an error. That error we cannot explain. That is called as experimental error, right? So in the last course also I explained what is experimental error. It is the error that occurs in experiments where we are unable to explain those errors. So that is called as experimental error. These are unknown errors. What do you think when you do an experiment? Would you allow more to happen unexplained error or experimental error, or would you like to reduce the unknown or the experimental error? What is your opinion? We say experimental error is happening, we cannot avoid, but the component, the size of the experimental error, what is happening? Why we want to reduce the experimental error? Can you remember we have talked about I'm sharing the screen. Can you remember we have done something like this? In the previous course, we have done can you remember? What is that? This is. Can you remember? Have you seen similar kind of thing in previous course? Yeah. What is that? Can you all see what I have written? I don't hear anything. Are you all alive?
Yes, sir.
Yes, sir.
Can you see the screen? What I have written?
Yes, sir. Yes, sir.
Ah.
Yes, sir.
Tell me what is that?
Yes, sir.
What is that? What is in the numerator? What is numerator? What is that?
Regression.
Yeah, regression. What is this? This part, what is this?
Mean square.
You are right. Once again. Once again, what is it? Once again, what is it? Mean
square.
Mean square, exactly. So you
Mean
see,
square.
yeah, regression mean square. You have seen this earlier.
Square.
Divided by what is that?
Residual mean square.
Exactly, residual mean square. You see, always when you do an experiment, how we can express this? Regression mean square divided by residual mean square. Oh, this f is calculated as explain variability. Explain variability. I will simply say explain variability divided by unexplained variability. Okay, explain variability divided by unexplained variability. So among the total variability, if you add these two variability, it is basically total variability, right? Total variability. If you add these two, it is total variability. So regression variability divided by variability due to unknown or residual variability. That is how we quantify. So here, this is the general formula. Explain variability divided by unexplained variability. So suppose, let's say, if the proportion of unexplained variability increases, what will happen to this F value? In an experiment, if you suppose to increase, without knowing to your knowledge, if the unexplained variability increases, right? What is happening to this F?
Decrease.
Reduce.
Decrease, right? If F decrease, what will happen to? I just erase it. If F decreases, so F is going to decrease. If F decreases, what will happen to the decision making process? Can you remember this decision making process? Sorry. These are the technical problems. Just a Uh, Sasi, so right, fine. So suppose let's say we have the value f, right? We said the f is going to decrease. If the f is going to decrease, what is your decision making process? Can you remember this term? Have you seen something like this? What is that? Have you seen something like that? F, C. How do you call this? How do you call this? How do you call FC? How do you call this FC? F calculated value. If F calculated value is greater than F table value, what is the decision making process? What is the decision making process? Rejecting or accepting null hypothesis?
Rejecting.
Yeah, reject our null hypothesis. Right? So that is our rule. So you reject our null hypothesis when the calculated value is higher. Similarly, if F calculated value is less than F table value, F calculated value is less than F table value, then we accept our H naught. Something goes. Right? Just a second. Right. If you accept the H naught, right? So you simply accept the H naught. If the calculated value is less than the table value. So that is the thing. Right. Now, what is our problem here? We said we accept our H naught or null hypothesis value if the F value is lower. So if F value is if calculated value is lower, basically, right? So we simply accept the H naught. We accept the H naught. So if you accept the H naught, what is our H naught? No treatment effect. So that is our null hypothesis. Alternative is there's a treatment effect. So there's a treatment effect. So the null hypothesis is simply says no treatment effect. Alternative says there's a treatment effect. Okay. So our problem is if the calculated value is lower, we come to a conclusion accepting the sentence that there's a no treatment. But unfortunately, even in case if there is a actually treatment effect, this treatment effect would be masked by this unexplained error. So this treatment, even if there is a treatment effect, this treatment effect will be masked. So that is the problem that arise with the unexplained error. So that is why we want to reduce the experimental error. So this experimental error occurs randomly without knowledge of anyone, but we want to reduce that experimental error as much as possible. So we are going to see how we will reduce the experimental error. Right. So this is the way that we can reduce the experimental error. Just a second. Sasi? Can you all see the presentation? Hope you can
Yes,
see.
sir.
Yeah. Okay.
Yes,
Fine.
sir.
So now we are going to see how are we going to reduce the experimental error. So the same thing which I have already shared with you. Right. Now, how do you call this? What is the play of this? Anyone who knows the name of it? This usually. What is the aim of this playing? You have to throw this this particular bow. This arrow has to be thrown to the middle of this eye. This is called as bullseye. Right? Can you all see my face?
Yes, sir.
Right. Okay. So the aim is throwing this arrow to this bullseye. Right? So that is basically your aim. Suppose. So bullseye represent the true value of the parameter that you wish to estimate. Can you name one estimate? Can you name one estimate? Miss Tasneem Banu? Is she there or AG 037 12 AG 12, right? Is Banu is around? Is Banu is around? Now it's your time. Banu is around. If you don't answer, then I'll have to pick off your name, saying absent for the day, because these are counted. These attendance are counted, right? So Miss Banu, Tasneem Banu, is she around? You can answer, no problem. So Banu is absent. So if I tell all the names, all will be absent, I think so. Huh? There are 74 names are there. So all of you are going to be absent, it's look like. Because no one is going to answer. If you all answer only, I can understand that you all are online. Otherwise, you can switch on and sleep. No problem. As you do in the class itself. Banu, are you around? This is the last call. So Banu is not around, right? So I'm going to make a make a point that Banu is not there. Right. What about Priyanka? I will go in a sequence order.
Yes, sir.
Priyanka is there. Your AG number is 97, right?
2015 AG 97.
Right. Now, Priyanka, tell me, what is the estimate? Because this is a continuation from the previous course. I have mentioned about estimate. What is estimate? Just tell me. Are you Tamil or Sinhala?
Tamil.
You can answer in Tamil also, no problem. I can explain in English to others. Yes, Priyanka. What is estimate? If you wish, you can answer in Tamil, right? Otherwise, you can answer in English. What is estimate? For example, if you want to, yeah, tell me first, Priyanka. You can answer in any language, yeah? No, don't worry about it. If you don't know, just tell me don't know. Then I will go to the other one. Yes, Priyanka. What is estimate? We have done a lot of estimates in our class during last semester. Suppose,
Exams.
Yeah.
Exam assessments.
Not really. Not really. For example, let's say in a class, how many members in your class?
74.
74. Now I want to find out average height of all these 74 students. So
Yes.
this is a small class so that I can simply measure the height of all 74 students. Suppose, let's say, if you have a very big class where I cannot take the measurement from all individual members, I will take a sample, right? I will
Yes.
take, even in your class, 74 members, I take 10 sample. If I make a mean or average height from this 10 sample, it is actually called as estimate, right? This
Yes.
average from sample, not from population. We know what is the difference between population and sample, right? So average height from a sample is called as estimate. So samples are always estimate. For example, sample mean is an estimate, right? Sample variance. Have you learned this sample mean, sample variance? Yes?
Yes.
Yes or no? Akilan?
Yes, sir.
Akilan is there? Akilan, not one.
Yes, sir.
Akilan, did you learn all these sample mean, sample variance from previous class? That is the first class that we talked about. Can you
Yes, sir.
remember? Yeah, all these are called as estimates, right? So if you take the sample, if you put up or calculate the mean value, or if you calculate, for example, variance value, or if you calculate the standard deviation, all these are estimates. Clear? Right. Now everybody should know what is estimate. So estimate, we want to find out estimate. What is parameter? Parameter mean it is the population concept. So this 74 students is a population parameter, right? So average height of this population, what your class 74 students, is actually how we indicate this parameter population mean. How do we indicate population mean?
Mu.
Mu, exactly, mu. So mu is a parameter. Mu is a parameter. That means population mean is a parameter. How do you symbolize the sample mean?
X bar.
Exactly. So X bar is an estimate of mu. Mu is a parameter. Is it clear? Mu is a parameter because it is coming and represent the population. So that is parameter. Mu is a parameter, population parameter. Estimate is the sample mean, which is called as estimate, right? So this is the difference between estimate and the parameter. Right, now we will come to the business. Accuracy. Accuracy without bias. What do you mean by bias? Simple example. Anyone? What is bias? Giving priority to somebody is a bias, isn't it? If I want to select 10 students from your 74 student group, I select randomly without giving any preference to anyone. For example, if I select only handsome boys and beautiful girls, do you think is it biased to measure the height? Yes or no? Ayora, not to?
Yes, sir.
So my question is, if I want to measure the average height, so that I have taken 10 samples from your class, now I'm selecting these 10 students by looking at their face. Boys, if they are smart and handsome, girls with beautiful faces. If I do select those samples, do you think it is a biased or non-biased? Or unbiased? Biased, because we are giving a priority to the beautiness or the handsomeness, isn't it? So that is called as bias. All right. So we'll have to maintain unbiased. So in other term, we call it as without any biasness, right? Okay. So accuracy is coupled with biasness. So accuracy is depends on if you are doing the work unbiased. So there's accuracy that we can assume. So average is on the bull's eye. So if you take the average on the bull's eye, right? If you take the average on the bull's eye, so this is the bull's eye, right? So if you take the average that you throw, then this is will represent the accuracy achieved through randomization. So this accuracy, that means it's related to the unbiased estimates that can be obtained using randomization. Can you remember what is randomization? Yes or no? Huh?
Yes, sir.
Yeah. So randomization, we assume, I mean, we learned this randomization last week. So randomization is important, right?
Yes, sir.
Okay. So accuracy is one thing that we want to achieve by doing that. What about others? I have problem. Yeah. Sorry. Second one is precision. Two different things: accuracy and precision. Precision mean, can somebody tell me what is precision? Say you are doing a titration, right? You are doing a titration in chemistry lab. My endpoint gives the reading from the burette is 5.2 milliliter. Have you all hearing me properly?
Yes, yes.
All right. Okay.
Yes,
Okay.
sir.
My question is clear, right? So I'm doing a small titration. Now my first endpoint is 5 point, what I said, 5 point, what I said, yeah,
5.2 mil.
okay, 5.2 milliliter. My first reading is, I did the calculation, my first reading is 5.2 milliliter from the burette, right? My second reading is, I take 3 readings. Let's assume I take 3 readings. So the second reading is 5.6 milliliter from the burette. Again, I am doing titration. So I got 5.6. The third one, I got 5.8 milliliter. That is one experiment. In another experiment, you see another separate experiment, I got 10.2 milliliter. These two are two different experiments. Right? Then as the second reading, 10.2 milliliter. Clear? The third one, 15.3 milliliter. So now can you compare these two and tell me? Can you compare these two? Tell me. What about the first experiment? So this is the first experiment, right? This is the first experiment. Can you tell me related to first experiment and the second experiment?
In first experiment, measurements are close together.
Yeah, more measurements are close together, right? Their differences are much less. In second experiment, yes. You see, these two are more or less okay,
Close
going
together.
close together.
Third one is yes.
Suddenly, you see the third observation, something wrong.
Yes.
Something wrong. This is where we come to a point. All right. This is where we come to a point, precision. So in our case, precision is repeatability. So we have repeated our titration. So in first experiment, more or less all the 3 readings are close together. Measurements are close together in first experiment. In the second experiment, you see last observations totally deviating, right? So what do you think about the experiment 1 and experiment 2? Which is having more more precision? Is it
First one is
experiment
low.
1 or 2? Yeah,
Experiment 1.
experiment 1, exactly. So this is what we call as repeatability. So based on that, can you recall the other principle of experimental design that we learned last week? Replication, isn't it? So other one is replication, right? Sorry. Right, replication and randomization. So this is relating to the control of experimental error. Don't forget that. If one do an experiment, both accuracy and precision are important. Both are important. You cannot throw one away. So accuracy as well as the precision, both are important. Right. Look at this diagram. Here we have sorry. Here we have precision and unprecision or imprecise. We don't call it as unprecise, imprecise in English, right? Precise and imprecise. Similarly, biased and unbiased. We already have seen these two terms, right? Biased, unbiased, precision. So opposite to those two words is called as unbiased and imprecise. Let's look at some schematic diagram. Can you all see this? Yes?
No, sir.
Huh?
No, sir.
No, sir.
Can't you see this slide?
No, sir.
Then,
No, sir.
No, sir.
No, sir.
then why you all are not telling me exactly? Can you see now?
Yes, sir.
Yes, sir.
Yes, sir.
Yes. Right. Okay. Right. Okay. So we already talked about two things. Can you see now? Yes.
Yes, sir.
Okay.
Yes, sir.
So we talked about accuracy. That is coming through randomization. The other one is called precision. That is coming through replication. So the measurements which are coming close together, that is coming through replication, right? Randomization and replication. So both are equally important. That is what I said. Now we have precise and unbiased. Precise and biased. So opposite to that is imprecise and unbiased. So this is how we can explain precision, unbiased situation. So this is precise and unbiased. Okay, we call it as precise and unbiased. This is precise but biased. So precision means repeatability, isn't it? So when you repeat it, it is coming in very close proximity. The values are much closer. So that is precision. But this is unbiased. This is unbiased. But in this case, it is biased. Why? It has gone. Rohini, what is happening? Everything I can see here. Rohini, Lakmini. Who is Rohini Lakmini? I have the list. No one can escape. Okay, doesn't matter. Hello, can you all hear and see?
Yes, sir.
Right. Okay. So precise, but this is biased. Precise means all the points are close together, but it is biased. Why? Our aim is to throw a arrow into the bullseye, but it has gone away from bullseye, right? Rovini making problem. Right. Okay, so the second one, this is unbiased but imprecise. Right. I don't know who is making all sort of problem. Who is Rovini? Lakmali. How is it? Lakmali, age number 116. Are you in or out? I have all the record, huh? Rovini. Yeah, can you all see me?
Yes, sir.
Yes, sir.
Ah, right.
Yes, sir.
Right. Okay. So please don't disturb the people by unmuting your microphone. Don't forget that all of you, when you log in, log out, all information we can get from one printout, right? Everything is recorded here. So we'll have to have some dignity, so that all will be recorded and taken out. How many people, I mean, who are the people logged on at what time, logged off at what time. But unfortunately, physically, we can't see whether you are sitting and listening to the lectures, right? It's up to you. Right. So this is imprecise, because our aim is to throw an arrow into this bullseye, but it has gone everywhere. But it is unbiased. It is unbiased because it is not focused on one place. You see, this is imprecise and unbiased. So our aim is here. So it has gone out of the aim, so that it is imprecise and biased. It is towards only on one direction. So nothing here. So it is imprecise and biased, right? Always, please remember, always you see a writing board or the PowerPoint. If you don't see any of those, right, just let me know. Right, I will switch on from PowerPoint to board, or PowerPoint board to PowerPoint. If you should see at least one of these. I mean, at least mean you should see one of these always. If you don't see, just let me know. Right? Clear? Tanushan, clear?
Yes, sir.
So I will catch up only Tanushan, because he's the batch rep, no? Right. Okay. So you'll have to see either of these, either whiteboard or this PowerPoint presentation. Right. Now, what is the importance of controlling variability, unexplained variability? This slide, which I have already talked to you earlier in last semester. Variabilities, it is the difference between the average value and the object of the data. So this is the deviation, how much it is deviate from that is called as variability, right? Can you all remember?
Yes.
What is variability? How? What is the formula to variability? What is the formula to variability? X minus X bar, right? What is coming in front? That is variance. So this is basically variability, right? So this is what we call as variability. Can you all see? Can you all see the board?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Right. Okay. So this is what we call as variability. It is the deviation from the mean value to each and every object's observation. So that is called as variability. So this variability, for our easiness, for example, how do you call this? It's two,
Sample variance.
sample variance. Yeah. How do you write? Anyone? Sample variance summation xi minus x bar,
X bar
whole thing
Right into
n minus
n minus 1.
n minus.
1.
Okay, right. So this is what called as variability, but we have learned long ago, right? This is how you calculate the variability. Right. So I'm not going to talk about variability further, since you all are very familiar to variability. Right. The same example that I have put forward in your previous lectures. Let's take 16 smaller similar tomato plants, right? 16 similar tomato plants, pots. It has been put in pots. 16 pots. In each pot, you have single plants, right? So to have a control of all the other variation, what we have done is we have taken these tomato plants from same, what do you call, same?
Variety.
Yeah. So if there's a same variety, what we can assume? What we can assume is there is a variability in genetic?
No, sir.
No.
No. To assume that there is no variability in genetic. They are from the same seed source, single fruit, assuming all are genetically same. Same potting mixture is same, and management also same. What do you think? Basically, all are same, isn't it? Since all are same, we can assume almost all the plants are at the same condition, no differences at all, right? Now what we are going to do is we are going to allocate 4 treatments. These are the treatments giving 5 gram, 10 gram, 15 gram, and 24 grams. These are the treatments, fertilizer treatments, right? For each treatment, we have chosen 4 plants, right? So now I'm going to measure the yield of all those 4 plants. Measure the yield of all those 4 plants. So what are the treatments? 5 gram, 10 gram, 15 gram, and 20 gram. So how many plants were there originally? 16 similar plants. So if I want to give 4 treatment, for each treatment, how many plants have been allocated?
4
plants, exactly. 4 plants have been allocated. So now I'm giving treatment. Ultimately, I'm measuring the yield. I'm measuring the ultimately the yield. So now you see these are the results of 4 plants that has been treated by 5 gram of fertilizer, right? So can you all get the same yield? So no, why? Because I said all these plants are similar plants, isn't it? Or just a second. You see all similar plants. So use so all 16 plants. Sorry, all 16 plants. You see, all 16 plants are similar plants. That is first thing. Second thing, we have taken from same variety, same seed source, same potting mixture, and same management. Same management means we add same amount of water, we do the weeding at same level, everything is same. But you see, suddenly, how many plants have been treated with 5 gram? 4 plants. If you measure the yield of all those 4 plants, the yields are giving different values. Can somebody explain what is the reason behind? Can somebody explain what is the reason behind? Tanushan, AG 037. What is the reason behind? We have 4 plants treated similarly, including the fertilizer as well, but the results is different. 037, is he Tanushan? K.
Yes.
I don't hear Tanushan.
Yes, sir.
Yeah. So what is the reason, Tanushan?
Hello.
I don't hear Tanushan properly. Oh, you can send me a message. You can send a chat. Another chat, you can send it. I don't get.
Okay.
Yeah. Tanushan can write to me, no problem. Yes, Tanushan. Your voice is not clear to me. You can chat. You can give the answer.
Yes. Yes.
You can write. What is your answer? Yes, I got Tanu. Fertilizer different. Are you sure? Because that is not for fertilizer different. Because I am saying we have given the same fertilizer. You see here in this point, we have given the same fertilizer, not different fertilizer. These 4 plants treated with 5 gram. You see,
Yeah.
5 gram,
Yeah.
not 10 gram. So,
Yes.
what could be the reason?
So different variety.
Not really variety, because all same variety. Here variety is same. All other aspects are same.
Yes,
You can't say,
same
huh?
variety. Yes.
Yeah, that is the important part. Because everything is same, but they are performing differently. So this is what we said, unknown. We can say unknown. I cannot write. It is basically, right? It is, we can simply say, because you see, I am simply drawing that. Here one reading, another reading, another reading, another reading. This is for 5 gram, right? So there's a variation. Actually, there's a variation, isn't it? There's a variation. So this variation is actually unknown. Unknown variation. So usually I used to say it is only God knows, nobody else. So this is the error that we are calling it as. This is the unknown error that is called as experimental error. This is the unknown error that we call as experimental error. Okay, even if you treat all the objects in a similar way, but the error is there. So this is called as unknown error or the unexplained error. Right. Similarly, this is true for the second type of treatment, that is 10 gram, 15 gram, and 20 gram. Right? 10, 15, and 20 gram. If you go for 10, 15, and 20 gram, all right, always this is the problem. Yeah. Let to close this, otherwise don't get. Yeah, sorry. So you see here, there are two directional variability, right? There are two directional variability. One directional variability. Can you all understand what I mean by two directional variability? You see the variability, if you go in this direction, this is one variability, right? If you go this direction, another variability. You see there's a this direction, upper direction, this is variability, isn't it? There's a variability. This is the variability within the group. Do you understand? This is within the group. Can you all see? Can you all see, please tell me? One person is
Yes,
enough.
sir.
Yeah.
Yes, sir.
Yes.
So you see there's a variability happening here.
Yes, sir.
Within the group, there's a variability. There's a variability here. So this variability is there. Right? How do you call this variability? Either this or this. This is unknown. I will put it as experimental error. I will put it as EE. Difficult to write. Experimental error. Right? Can somebody tell me there's a variability increasing in this direction? How we can explain that? That is because of increase in fertilizer. Normally, as you all know, when you increase the amount of fertilizer, what will happen to the yield? Starting from lower value, yield will increase. So that variability that we can explain. So this variability from this point to this point, this point to this point, there's a variability. That variability is due to increase of fertilizer. Isn't it? The fertilizer increasing gradually, 5, 10, 15, 20. So that variability we can explain. So I will put it as EX. That we can explain the variability within the group, within the treatment group, that we cannot explain. That is experimental. Is that clear? Right. Now, if you come to, sorry, I have again, yes, this is experimental error. So in the experimental error, we can, the total variability, we can partition into two groups. Usually when we apply treatment, it will increase the variability. In our example, what is the treatment? In our example, what is the treatment? 5 gram, 10 gram, 15 and 20 gram. What is the treatment?
Fertilizer.
Exactly. So the treatment, when the treatment increases, the doses of that treatment increases, it increases the variability, right? That can be assumed. When the treatment, I mean, usually when you do an experiment, right? When you do an experiment, when you do experiment, let's say this is initially I have some weights, some objects, right? Weight, let's say weight 50 kilogram, 28 kilogram, 32 kilogram, 42 as an example, 42 and 55, right? If you look at, if you calculate the variability, let's say V1, can you calculate the variability? Yes, we can calculate the variability, right? Then I am applying a treatment, right? I am applying a treatment. Let's say, what is the treatment? Hormone treatment. Let's say hormone treatment. Hormone treatment. I will put it as HT, right? I will put it as HT. So this HT treatment, after this is before giving the hormone treatment, you have the weight, right? So after treating with hormone, we are measuring the weight. Let's say I am not going to put any value. I'm going to put it as X. So A, B, C, D.
Sir, could not hear your voice clearly, sir.
Now, can you all now?
Yes, sir.
Okay,
Can you
sir.
all?
Yes, sir.
Yes. Okay. Right. Now you see, you can see the board, right?
Okay.
Yeah.
Yes, sir.
Right. Yeah. Okay. After treatment, what I did is I have measured the weight. After hormone treatment, I have measured the weight. I didn't put the actual figures. I'm simply saying A, B, C, D, and so on. This is before. What is this? This is before hormone treatment. This is after the hormone treatment. Now I am measuring the variability. So here also I measure the variability after the hormone treatment. Here I take it as V2, right? If there are really actually effect on weight by hormone treatment, do you understand? If in reality, if there is an actual effect on body weight by the hormone, then the variability, if you compare the variability V1 versus V2, what do you think? I said there is a real effect by the hormone. What do you think? He also, because same object, because this is from the same person. Initial weight is 50 kilogram. After hormone treatment, A, right? Maybe this hormone is to increase the body weight, let's say, to increase the body weight. Huh? These people are underweight people. Let's say there are 5 people, underweight people. So this underweight 5 people have been given treatment, hormone treatment to increase the body weight. Let's say A, B, C, D, E are the respective value or increase of the body weight. Now my question is, we can measure the variability separately for these 5 data before treatment. We can measure the variability for these 5 data as well. Clear? So those variability, I simply set V1 and V2. V1 and V2. Now can you tell me, suppose if this hormone is really good in increasing body weight, suppose, because we are doing an experiment. So if the hormone is really good, that could have been increased the body weight, isn't it? Do you agree? If the hormone is good, then it could have increased the body weight. So now I have measured V1 and V2. Can somebody tell me which value is higher? V1 or V2? Which value is higher if there is an effect of hormone? I will put it as V1, V2 here. So V1, V2. Can somebody tell if this is correct? Which one is correct? First one or second one? Which one is correct? Which one is correct? Why I am saying is that look at this term. Treatment will increase the variability. So if there's actually treatment, it will increase the variability. So based on that, can you tell me precisely which one is correct? First one or second one?
Second one.
Second one.
Yeah,
Second one.
second one, exactly. So the correct answer is this. I say if there is a hormone effect only. If there is a hormone effect, then we can say the variability due to treatment has increased, right? So that is our conclusion. So you'll have to be careful. So this is how we are going to measure this variability after the treatment and before treatment. As an example, we compare the variability between these two and see whether it has increased or not. If the variability has increased, then definitely we can say there's a treatment effect, right? So we can understand that later. We will look at further. So in that respect, sorry, this computer is very slow. So what we measure as the response at the end of the experiment is total variability. So total variability what we measure. So total variability we can partition into two groups. One is called as treatment variability, variability due to treatment. The other variability is error. So this treatment variability can be explained. That part of the variability that we can explain. Treatment variability, that part of the variability that we can explain. But the error variability, that we cannot explain. Error variability we cannot explain. So that is the drawback of this variability concept. So you see, when you have total variability of one particular experiment, we have explained variability, we have unexplained variability. According to your knowledge, which portion has to be high? Whether it is explained variability or unexplained variability. Explained variability. So that we can explain by experiment. In the early experiment, for example, we can explain due to the increase of fertilizer. When you increase the fertilizer, yield increase. That is logic, isn't it? So that we can explain. But when you treat with the same treatment among the objects or among the plants, the yield that we obtain is different because we treat everything similarly, but we obtain different results, right? That is called as unexplained variable. In the previous example, I took titration. Titration. Usually in chemistry, titration, it is not very that difficult. If you take, say, titration, in titration, similar to early example, in one experiment I got burette reading 5.5 milliliter. Can't you see? Can you see?
Yes, sir.
Okay, can you hear?
Yes, sir.
Okay, right. So this is 5.5 milliliter. This is 5.5 milliliter. I'm doing again, repeating the same titration. I get 5.7, 5.7, right? The third one, 5.6 milliliter. Right? So now, can you tell me, this is also milliliter, can you tell me why are we getting different answer? Why are we getting different answer? Assuming that no human error, no instrumental error, no other errors. Assuming that no other errors. Same person is doing the titration. Have you all done some examination titrations in the examinations?
Yes.
So have you ever got same results? I mean, when you do titration 3 times, have you got same results or different results?
Different results.
Different results.
Why? Why? Because 5.5, right? In this case 5.5, this case 5.7, in this case 5.6. This is, here there's a variability. This variability, this variability, we cannot explain this variability because all are treated similarly. The variability that we measure here, we cannot explain. So this is called as unexplained variability. Is it clear? Assuming that all other factors are constant. Same person is taking the reading, same beaker that you are using, same burette that has been used, same chemical mixtures that you are using, everything is same. If you use a different chemical mixture, the value might change. Why? Because sometimes you might have diluted with higher value, maybe 5 milliliter more or 5 milliliter less. If you mix it with some chemical, definitely it will show the end point as not equal. But in our experiment, everything is same. So what is the problem? So this is called as unexplained variability. Right? So ultimately, the total variability that we can say explained variability, unexplained variability. Right. I'm coming to the same point again, which I have already discussed. So what is wrong with this unexplained variability? So in the hypothesis, null hypothesis, usually we take no treatment effect, right? We take no treatment effect. All right. I will simply say no. No treatment effect. Here, there's a treatment effect. Alternative hypothesis, there's a treatment effect. So now we go for test statistics. Test statistics, this is what I said earlier. The calculated value is based on the mean square that you have explained. Treatment mean square. Treatment mean square. Isn't it? Divide by mean square of error. Error mean square. So always, how did we call this component with another term? Can somebody? Yeah. Nirupama, yeah. I don't hear it.
Total variability.
Total variability. If you add this variability plus this variability, that is total variability. But how I'm asking, what is this portion? What is this portion?
Regression error.
Regression.
Regression error.
Regression mean.
Yeah, I mean, in general, it is called as explain variability. Explain variability. This we can explain. variability that we can explain. So how do we call then this one?
Unexplained variable.
Exactly. So this is what we already said. You have the notes, ne? Notes langapadanan. Don't you have notes? This is also now not working. See. So this is what I want to say. Explain variability, divide by unexplained variability. So these are the two variability that we are talking about. So the decision making, as I pointed out earlier, is totally depend on this F calculated value. Right? It's totally depend on this calculated value. So if the calculated value is lower, then we do not reject our null hypothesis. If the calculated value is higher, then we reject our null hypothesis. Okay, which I have already discussed. Right. Control of unexplained variability. What are the factors that we'll have to consider? First one is experimental error. Sorry, experimental side. So when you do experiments, for example, the people who are specializing in agronomy, maybe animal science, doing with pasture, maybe chemistry, maybe with other biology, when you do culture, etc., you'll have to, because experimental site where we are doing, maybe are we doing in the field, or are we doing in the plant house, or are we doing in the pots, or are we doing the experiments in the petri dishes, right? So that is called as experimental site, where we are going to have it, all these experimental units. Last time I explained what is experimental units, isn't it? So how are we going to, and where are we going to have this experimental unit? So based on that, we can have our experimental site. It is a place where we do the experiments. Could be plant house, could be tissue culture lab, or could be the field, or could be the field closer to the river, or field closer to the road, right? All these are matters. So based on that, we will have to decide whether the site is homogeneous or heterogeneous. We discuss about these two terms. What do you mean by homogeneous and heterogeneous? In the laboratory, for example, from one corner to another corner, if you walk, do you feel difference in temperature, difference in light, as an example, in a laboratory?
No.
No, usually not. So that kind of field or environment, how do we call?
Homogeneous.
Homogeneous, exactly, homogeneous. The environment is more or less homogeneous. The other type is called as heterogeneous. Suppose, let's say, soil. If you take soil, typical example, everybody knows what is soil. Suppose if I take a soil sample where I am staying now, I will walk 10 meters away from that sample taken, and again doing a soil sample in 10-meter distance. Can you comment on these soils? Are the soils difference in physical chemical parameters, or will it be same physical chemical characters? For example, cation exchange, amount of nitrogen in the soil, amount of phosphorus in the soil. What do you think?
Not same.
Not same exactly. So the soil basically heterogeneous condition. We cannot consider soil as a homogeneous. For example, let's say the soil in a slopey land, soil in a slopey land, right? Yeah. So the land. So this is a slopey land, right? So I'm taking soil here. Example. So this is sample one.
Could not hear, sir.
Now,
Okay, sir.
Soil sample one. Let's take soil sample two. Soil sample three. These are slopey land. Can you comment about the fertility of these samples?
S1 is less fertile than the S3, sample 3.
Yeah. What about if I write like this? Do you agree?
Yes.
Yeah. So S3 is high fertile compared to S2 and S1. Why? Because as you all know, here less topsoil because of the heavy rain. It is eroded, topsoil is eroded compared to here. Here it is very high with fertility. Why? All the soils have been brought down here. Isn't it? Right. So this is as an example, this is a typical example. So in the soil, you have a heterogeneous condition. Right. In the soil, you have heterogeneous condition. This is what I said, gradient. In the slope, we'll have to do the blocking. We will come to that, what it is later, one-way blocking or two-way blocking. Then experiments with animals. Last time I have mentioned about, could you remember, we cannot take same weight animals. Maybe with age possible, breed possible, but we cannot make animals, take animals with the same weight. So we have problem. So how we can decide all these? Because all these will contribute to the unexplained variable, right? Then the other one is type and number of treatments. So what type of treatment are we taking and how many treatments are we taking? Later we will see when you do can you all see me? Can you all see me?
Yes, sir. Yes, sir.
Yeah, okay. Board you can see. So later we will see because this is called as block. I'll simply say BLC block. BLC BLOCK. I will simply say block. So each of these are plots, right? Each of these are plot. Maybe when you do experiments, this is 2 meter, for example, 2 meter plot. This is also 2 meter plot. So likewise, we can have. So this is here we'll have to assume there's a homogeneity. Then only we can apply different treatment. For example, treatment 1, treatment 2, and treatment 3, and a treatment 4. 4 treatments we can apply. So each of these are same size plots. These are called as plots. Right? So now, if you have large number of treatments in early case, for example, this is called as complete block design. CBD mean complete block design. IBD mean incomplete block design. You can write down complete block design. IBD mean incomplete block design. All right. So you see now, if I have suppose if I have 4 treatment like this, no problem. Now let's say I have 20 treatment. I'll put it as treatment. Starting from T1 goes up to T20. I will just name it as T20. Now I want to have a very large block, isn't it? Well, this is one plot, second one. So likewise, how many I need? How many plot I need to accommodate all 20 treatment? Anyone? To accommodate all 20 treatment. Yeah. Tanushan P. How many plot we need? Anyone? Like here, how many treatments are there? Here, how many treatments are there? 4. So how many plots we had here? 4. So here we want to accommodate 20 treatments. So how many plots do we need? 20, exactly. So we need 20. Now when you have 20 plots here in one set, what is the problem arise? So here, if you do the soil sample, for example, and if you do the soil sample at the last plot, let's say this is the last plot. What is the total distance? If it is 2 meters each, from this point to this point, what is the distance? One kilometer. If the distance, I mean, if the plot width is same 2 meters, what is the distance from here to here? Anushan,